Hey guys, here we are at section 3.3, the plasma membrane, and we're going to discuss how substances get across the cell membrane. To do that, we need to talk a little bit about the structure of the plasma membrane and the types of molecules that can cross it, distinguish between diffusion, osmosis, and facilitated transport, explain tonicity, compare and contrast passive and active transport, and then we're going to summarize how eukaryotic cells move large molecules across the membrane. The characteristics of the plasma membrane start with the phospholipid bilayer. I want to remind you this is built out of lipids and it's in two layers. These lipids are embedded with proteins that can move around in space. There's cholesterol for support and carbohydrates on proteins and lipids. This is a selectively permeable membrane. And what this means is if you look at this phospholipid bilayer here, along with these embedded proteins, the carbohydrates that are hanging off the end of that, you can tell that there are little tiny gaps between these, and some substances can easily pass through this membrane. Others have to go through these channels, and some are just flat out too big or too charged in order to get across the membrane itself. And so this selective permeability allows the cell to have some control over what's crossing the membrane. So you can see here a large macromolecule is too large to cross that membrane right here. Non-charged molecules can cross very easily. And charged molecules are actually repelled. And so you would have to have pumps or channels in order for those to move. The 5 key ways that things move across the plasma membrane are through simple diffusion, osmosis, facilitated transport, active transport, and then endos and exocytosis. And we'll discuss each one of these. Diffusion and osmosis are the simplest. We simply take advantage of the fact that molecules are randomly moving at all times. And this movement tends to spread them out. If you start with a really high concentration, over time, that's going to diffuse out and spread out until things reach an equilibrium. The general direction of movement is, or the net gain in movement is from large concentrations to lower concentrations. Osmosis is diffusion, but it's specific to just water molecules. Here you can see a selectively permeable plasma membrane. And in this case, we're starting with more water on the outside of the cell and more solutes on the inside of the cell. And the consequence of this is over time, we reach equilibrium. Although water is moving back and forth across that membrane at any given moment, and those solutes are moving back and forth across the membrane, given enough time, they will eventually have the same number leaving as are entering, so that we reach this equilibrium. We can be more specific when we start talking about osmosis, and we talk about the tonicity, and we look at the comparison of the concentration of materials outside versus inside the cell. And if those concentrations are equal, we're going to call it isotonic, iso meaning the same, and tonic referring to the tonicity. So solutions that have equal amounts of solute inside and outside the cell don't typically have a big change occur. Although water may move in and out of the cell, not enough moves for it to really have any net gain or loss. However, when we deal with hypotonic solutions, they have less solute than the inside of the cell, meaning these solutions have more water per 100-gram scoop. Inside, you would have less water per 100-gram scoop. So again, we're comparing concentrations, not overall amounts. And you find that if the concentration of the water is outside the cell is higher than inside, then you will have water move into the cell, and these cells will become larger and larger and larger. In fact, they can lead to lysis or bursting of the cells. A hypertonic solution is the opposite situation where you have more solutes outside the cell than inside and less water per 100-gram scoop compared to inside the cell. And the net gain of water then moves out of the cell, and you see these cells shrivel up and shrink. The next type of transport is facilitated transport, and this involves molecules that are too large to cross the membrane on their own. So what we have is a channel protein that allows those molecules to cross. What's cool about channel proteins is they're exactly the same shape of that one particular molecule, so other things can't use that protein to cross. In this case, we just have sugar, where we have a larger concentration outside than inside the cell, and you can see the sugar is moving through essentially diffusion, but it's going through that channel protein. So that's why we call it facilitated transport. Active transport also uses a channel protein. However, we're going to move things against a concentration gradient. Naturally, things move from high concentration to low, but if we wanted to move it backwards, it's going to take energy to do this. That's why it's called active transport. And we're going to actively use ATP to get the energy to move things backwards to the concentration gradient. The next way we move things across the cell membrane are endocytosis and exocytosis. Endocytosis transports molecules into the cell, and exocytosis transports molecules out of the cell. Typically, we have something that is so large that it can't cross the cell membrane, or it's a package of a lot of molecules, like in a vesicle. And the way this works is that the plasma membrane itself engulfs material for endocytosis and brings it in using a vacuole, or there's a vesicle which has been created by like the Golgi apparatus where it's packaging materials, and that vesicle then can be transported to the edge of the cell. The membrane bonds with the cell membrane, and it can allow those materials to move out of the cell. Again, this is for much larger transport. So when we're thinking about how things cross the plasma membrane, we need to be thinking about the structure of the membrane, using diffusion, osmosis, and facilitated transport, all passive. And then we can think about the tonicity or the concentration gradients that things are going to move across. Then we have passive and active transport, and then endo and exocytosis. You guys did a great job. Thank you.